like to thank ChemAxon team for having invited me here. It's really a pleasure to be in Budapest. And I would like to tell you a little bit what I am doing at the Nestlé Institute of Health Sciences. We are a research institute founded by Nestlé 5 years ago, but I would say fully operational only since 2-3 years ago. We are based near Lausanne, in a nice place, and we have around 150, mostly biologists, but there are a few chemists as well. So the objective of our institute is to explore the link between nutrition and health. So we are all aware that there is a link. You know, now we have had coffee, or if we have chocolate, it stimulates us. Spicy food, like Hungarian paprika, will stimulate us. On the other side, the chronic consumption of unhealthy foods will also have bad consequences. But surprisingly, there is a little high-quality scientific studies that have been done to explore this link. And one reason that is actually not so easy to do them. So first, humans are genetically very diverse, unlike mice that are bred for experiments. And also, it wouldn't be ethical to kind of enclose humans in cages and feed them 6 months with a prescribed diet. But then the objective of our institute is to kind of look at this link at the molecular level. And we want to find molecules that originate from food, but that show some pharmacological activities that are basically bioactive molecules. And one way to go around it is to use high-throughput screening. So in order to do that, we need compound libraries, screening libraries, and high-throughput screening platform. So this is what I'm going to talk about, compound libraries. Now, because we are focusing on food, so we are limited to molecules that are found in nature, so natural products. And when I say this, I would define them as all non-man-made molecules, organic molecules, and not macromolecules. So first, let's look a little bit, how does this chemical space of natural products compare with the kind of typical molecules that are of interest in pharmaceutical companies, synthetic molecules. But before that, I will look a little bit at the size of the chemical space of natural products. So probably you remember that the estimated size of drug-like molecules is 10 to the 64, which is a huge, huge number. But then when we limit ourselves to kind of known molecule, when we look at the large public databases, there are now around 30 million molecules, so kind of we are inching toward 100 million. Typical big pharma will have around 1 million molecules. But then when we combine different databases of natural products and metabolize, then we're about 300,000, 400,000. I heard from Lutz Weber yesterday, he estimates it to 500,000. Okay, let's now look a little bit at how the natural products compare with synthetic molecules. So here I will quote a nice analysis done in the group of Giesbert Schneider a number of years ago. So of course, when you want to compare two things, first have you decide based on what you want to compare it. And when you compare molecules, like the maybe easiest thing is first to look at the molecular and physical properties. So what they did, they look at the Dictionary of Natural Products, which is a commercial database of natural products. And then they constructed a database of drugs and drug-like molecules, and then they calculated a number of properties. So although the size, average size is kind of very similar, especially when you remove these glycosylation chains, yeah, there are still some differences. First, the variability, which is shown here in parentheses, is typically bigger for natural products than it is for synthetic molecules, which means that kind of as these medicinal chemists tend to focus in a certain range of properties because they know these property ranges kind of work the best, while nature is much more kind of diverse there. Then in nature, we find more polar molecules, and that can reflect itself. They will have a higher number of hydrogen bond donors and acceptors. Also, log P will be lower, TPSA will be higher. Another interesting difference is the number of aromatic atoms. While medicinal chemists love aromaticity, so every second atom in an average molecule is aromatic. In nature, situation is different. It seems it's not so easy to synthesize aromatic molecules. similar for the number of kind of heteroatoms. And then another interesting thing is the number of chiral centers. So while synthetically they are difficult to treat, so typically avoided, nature doesn't have a problem with that. Another way to compare structures is to look at chemical structures. But because they're very diverse, some 20 years ago, Bemis and Merkel came up with a nice concept which they call molecular networks. So basically, they simplified molecular structures by keeping only rings and linkers between the rings. And this is what I call scaffolds. So this is very suitable for synthetic molecules because they often have ring systems. So by doing this, we can come with this atomic scaffold where we remove all the substituents unless they are connected by a double bond. And then in the second step, one can go even further and then mutate all atoms to carbons and all bonds to single bonds and make these graph scaffolds. And then kind of reduce the complexity of our set. So now let's look a little bit at the results. So this rectangular is kind of chemical space. So we see for the drug-like molecules, like only 2% are acyclic, meaning their scaffold is carbon atoms, so not very interesting. And then for these 100,000 molecules, we get about 50,000 different atomic scaffolds. So about 2/3 are scaffold singletons. It means there is only one molecule that has this scaffold. And then smaller number will have higher number of molecules. But then we will need about 5,000 scaffolds to describe half of the database. So this is this 11%. And then when we look at it for natural products, we have some more acyclic molecules, but we have fewer number of scaffolds, and we need only 1,000 scaffolds to describe half of the database. So basically, we can say that this is a bit counterintuitive, but actually there is higher scaffold diversity among synthetic molecules than among natural products. So now I wanted to focus a little bit on our in-house collection. So at the moment, we have about 20,000 molecules, and I could kind of repeat this exercise. When I calculated properties, basically, they were pretty much the same for our in-house collection and for the DNP. But then when we look at the scaffolds, then we got kind of the following picture. Okay, now we don't have sets of the same size. Ours is much smaller. But we have, relatively speaking, higher number of scaffold singletons, and we need, relatively speaking, more scaffolds to describe our collection. So it basically means that in our library, we see higher scaffold diversity, which is actually good because we plan to expand our library, meaning that we can add, other scaffolds and add maybe some other molecules that share scaffolds with these scaffold singletons. So this work was very easily done due to JChem and KNIME. So I want to mention some of my kind of favorite JChem KNIME notes. So when one deals with the collections of molecules, first one has to standardize them in order to be able to really handle them and compare them. And there I heavily relied on the Standardizer node. So it's a kind of procedure that was already mentioned in one of the talks yesterday. And then in some cases, I will also generate kind of molecules with deleted stereochemistry because I want to really kind of, in some cases, where I want to flatten them. So I want to thank the team for developing these useful nodes. Another node that I use a lot is chemical terms, where all these properties that are made by calculator could very easily kind of then be calculated. And equally, I could use the chemical terms to generate the Bemis-Merkel scaffolds. Another very useful feature, especially in the field of natural products, is the name-to-structure conversion. So natural products, it's kind of a field where people still call their molecules by name. And one finds itself in a tricky situation where you have like a list of very diverse names, and it's impossible to know them all. And then without this tool, it would really be tricky and would require lots of efforts to basically get structures for all these molecules. So I was then approached by one of our biologists who wanted to screen our screening collection. However, his assay is not a high throughput, but rather medium throughput, so he cannot screen all of them. So he asked me to make like a meaningful subset. So given our size, he needed about 15% of what we have in total. So then the question was, what would be a meaningful way to make a subset? One, you could say like a general diversity set, but maybe we can do a little bit better than that. So the set should be screened in the cellular assay, which means that molecules have to penetrate the cellular membrane. So you could say it would be nice if they are bioavailable. So that would be maybe one of the requirements. Then also, if biologist is limited to how many molecules he can test, then why not select molecules that have better chance to bind? And then we would like to keep the diversity. So we would like to keep representatives of all chemical classes that we find in our set. And in natural products, you have these flavonoids, alkaloids, terpenoids, etc. So these were kind of the three kind of guidelines that we propose for this application, to have like a bioavailability filter, something that I call like a best chance filter, and then to assign chemical classes to our molecules, and then to select diverse sets for each of these chemical classes group. So how to go about it? For bioavailability, that was kind of easy. We can just use the Lipinski, and I think everybody here knows, so I don't need to explain it. Then for the best chance filters, then we thought, let's maybe look a little bit what drives protein-ligand binding and look a bit at the basic thermodynamics. So as you remember, we have a protein and ligand. They're found actually in water. They're hydrated, and actually presence of this water is something that kind of complicates a little bit equations. And if we look in kind of step by step what happens in between, then we can dissect this process into following steps. So first, in the first part, we need to dehydrate our ligand and our protein. So we have to break the ligand-water bonds, which means if our ligand is nicely polar and charged, we need to bring a lot of energy to break these bonds. We need to do the same for the protein, but kind of whatever you need to bring here, you kind of get back here so you can ignore this step. On the other side, if you have a very lipophilic ligand, it will constrain water molecules around it. And these ordered water molecules will be all too happy to go into bulk water when you kind of get them away from the ligand. The next step, you have to constrain this freely moving ligand and the protein's binding site into binding concentration. So basically, we're going against the entropy. And then the final step, you will have if you have a good match between protein and ligand, then you will have a kind of good reward here. So then we could maybe summarize this, that what statistically leads in a good binding energy. So if you have unfavorable ligand hydration, if there is only low increase of entropy upon adopting a binding conformation, and of course, if you have a good protein-ligand interactions. However, in this case, we don't know what our target is because it's a cellular screen, so we cannot really address the last question. But then we could look for the kind of A and B, and saying if we have lipophilic molecules, so they will have a kind of good binding energy, but it could also be molecules that would bind to anything, so they would be non-specific binders. But then these very lipophilic will already be excluded with the Lipinski filter. But then we could maybe refine it a little bit and say maybe molecules that are highly planar, that have large number of, say, aromatic rings, they will also tend to kind of stick everywhere. So maybe these are the molecules that we can exclude in our set. Similarly, molecules that have very few polar atoms would kind of also fall into this category, even if they have a log P in a good range. On the other side, if we have very polar molecules, so they will be difficult to dehydrate. But there is another kind of obstacle here, is that they would have a hard time to penetrate the cellular membrane. So basically, they have low chance to get into the cell. So maybe these are also the molecules we can exclude. And then finally, molecules that are very flexible, that have large number of rotatable bonds, is also maybe molecules that we can exclude in this set. So on these two slides, I want to show you some examples of the molecules that were excluded. So all these filters are very subjective. I think any other person who would do this exercise would do it a little bit differently. And it doesn't mean that these molecules have no chance to be bioactive, just we believe that statistically they have less chance. And then, for example, we see stavrosporine, which, as you know, is a pan-kinase inhibitor, but I think skipping pan-kinase inhibitor, it's not such a big problem here. Then a lot of other, like, highly aromatic molecules. Some of these, which maybe would be interesting for the olfactory GPCRs, but these are not our targets here. Then again, here looking at some very polar molecule, like this is like a penicillin derivative, and as you know, penicillin has to be injected. It's not orally bioavailable, so will not go through the cell. And also some of the kind of highly flexible molecules. So to kind of, and then there is the third part. This was to select diverse sets for each chemical class. So now the tricky part was how to assign chemical classes. So one way would be to take a textbook on secondary metabolism, look at kind of scaffolds. How do you define flavonoids? Then draw all these scaffolds. But you know, who really wants to do that? Luckily, we have a dictionary of natural products in-house, and in this dictionary, we also have annotated chemical classes to which molecules belong. So basically, you can extract this information and then link it to kind of each of the molecules. And in this case, I generated kind of atomic scaffold for each of the molecules. So I would basically have like a relational database that would combine different atomic scaffolds with corresponding chemical classes. So by doing that, we ended up with 45,000 entries. And only 30% of entries would have multiple assignments, meaning that a particular scaffold would belong to more than one chemical class. So I think that's pretty fine. So, but now having this tool, we could finally assign chemical classes to all compounds in our collection, and then we could kind of do also some analysis and compare it to what we see relative to the dictionary of natural products. So this is kind of what we see here, the distribution. You start with aliphatic natural products, alkaloids, amino acids, peptides, et cetera, and you see that the kind of wheel is quite similar for our in-house compounds. We have less of the peptides, but actually these are not what we are interested in. And we have more of the flavonoids, because we inherited some molecules there. But kind of this way, this is the analysis that we could want to do. And now when we kind of look at this workflow, we could see that starting with 60,000 compounds, we could now basically filter it down to the requested number. And now another biologist approached me and says, I would like the same thing, but our project is a CNS project, so we would like our molecules to penetrate blood-brain barrier. So I have only 2 minutes, I have to rush, so we'll not explain too much about it. But there is a computational method developed by colleagues of Lipinski from Pfizer. They pretty much imitated Lipinski's approach by calculating different properties and identifying in which ranges you find most of the CNS drugs. So they came up with the CNS multi-parameter MPO score. And it took about 5 minutes to implement in JChem and KNIME. So thank you very much, the way to impress my colleagues. And then we could basically add an additional filter for the compounds for this project. And it was also nice to see that almost half of our collection would have favorable CNS score. So all of this work was possible due to a very nice kind of JChem package and the way it is very nicely merged with KNIME. So this is one example of my workflow that could very easily be kind of manipulated. So if you need a different size of subset, you can just change one number. If you need a different source, you could just change the link there as well. And for another project, biologists also asked me, oh, you know, because we are Nestlé, we do a lot with coffee and chocolate. Could you add some coffee and chocolate metabolites and see maybe some of these compounds could be active? So I could look in a dictionary of natural products and identify some compounds that are found, that are isolated from coffee or from cacao. Then I looked, I did the similarity search in our collection, and then I kind of expanded this set. And then finally, I applied the same filters I applied in the original set, and then I could kind of reduce the number. So I ended up with about additional 300 molecules. So this was also very simple to do in JChem with KNIME. And with this, I would like to thank many of my colleagues from the NIHS. I would like to thank ChemAxon team, and apologize for not including many more people to help me, and also John Fuller from KNIME. So thank you for your attention.
I will have just one very little question, because actually you are mentioning the differences between natural products and drugs. And then you made analysis of diversity in terms of scaffolds, which I would like to ask how justified it is, because since you showed that in nature you have much more polar molecules, I think if you just strip it to scaffolds, you are kind of losing this part of information. And I think scaffolds are maybe better suited, like for drugs where you have lots of rings and so on. So could you comment on that?
Mm-hmm. Well, first, depending which metric you choose, you will get a different answer. So, and I would say in a kind of drug discovery field, there is, especially in the recent time, a lot of compounds providers. They say, oh, you have to look for the three-dimensional molecules that are more natural product-like because they're more diverse. And then it was interesting to do this study to actually see that that's actually not what one sees. But it's true, like nature has ways to generate certain number of scaffolds, and then it decorates them in different ways. So it gains this diversity through these decorations. So you have these molecules that have, very diverse functions, but it's not only due to the scaffold.
I would say maybe if you say something about diversity and you say that in nature there is lower diversity, I think this is directly linked to the way how you do the diversity analysis.
Yeah, this is diversity based on scaffolds.
Yeah. Because if you base it on scaffold, you inherently favor like the man-made species. So I would say that's not really a fair comparison.
Yeah, exactly. So this is why on the slide it says based on the scaffolds, the natural
Sure.
products are less diverse. Yeah.
I was just curious, when you're talking about class assignments to groups of molecules, you say you have pre-annotated classes of molecules. Did you do some sanity check on how diverse the individual candidates you had in the classes against each other? So what's the diversity space you're covering by the pre-annotated classes? That's what I'm asking.
Okay, so when I did the diversity selection, it was then, I would then separate my molecules I would separate different classes, and then I would select, say, 20% from each of these small subsets. So whether with like 20% of flavonoids, 20% of terpenoids, etc. And there were also molecules for which I could not do the assignment because nothing matched, or they matched to these scaffolds for which you have multiple assignments, and then I just did 20% of those. So, yeah.